Yeah, thank you very much, Maurizio, for hosting us here. It's really a great city and a very inspiring atmosphere. I think this we can already say after having one day or even a little bit less here in Padua. And it's Ellen and my pleasure to welcome you now to the first official day, to the HIBALL workshop, which is number 8 already. And well, HIBALL is a German, European, Canadian, American, international, I would say now, consortium around the BigBrain, which is a project that Ellen and us in Düsseldorf and Jülich started many more years ago than HIBALL is existing. It took quite some years to develop the BigBrain model, and the BigBrain model was pioneering in many respects, starting from the computational side. You have used in Canada one of the first supercomputers with respect to histology. Wet lab, it was really something, I would say, like a Ferrari Formula One. So it's really this type of quality that is needed, and therefore it took really quite a lot of time to bring the different expertises together and really to form this network. Interestingly, when we started to discuss this idea, AI became more and more visible, and we both had the vision to use this model in order to inform AI, but also to use AI in order to analyze it. Because it became very clear, quickly clear that one brain with a highly folded surface is so big that you cannot just do it with traditional methods of image analysis as we have seen it. So that was one of the first data sets that we have released, and Claude did a lot of repair work for many years together with the team also in Jülich. And we reconstructed the BigBrain at 20 micrometer spatial resolution. It is still I would say, the highest resolution cellular model, histological model. And when we are cutting a brain, staining a brain, of course, we introduce artifacts which are visible in the orthogonal sections, but we are improving the quality also of this brain step by step and get really to an extremely high resolution. That are researchers coming from different labs, and we organized the HIBALL consortium in such a way that we have set up different work packages. You have them here, and we tried that every work package is represented by specialists coming from 2 different fields or from 2 different countries. So we are regularly meeting in these work packages. PJ and Susanne are really very strict in making sure that we have a very close collaboration. And at this point, I would like to say a particular thanks to PJ and Susanne for making this also possible. So I think the 2 ladies also in preparing this workshop did a great job. So please join me in saying thank you to the 2. Okay, but there's a lot of interesting research in. So there's more this neuroscience-driven research, I would say. This is work package 1, 2, 3. 4 is very special because our idea was also that we are building a transatlantic bridge to exchange the large data sets, and this requires tools very often based on supercomputing that speak, so to say, to both sides, and therefore Maurice Riedel from Iceland and Tristan Glattard from Canada are collaborating. And then also from the very beginning we thought structure is fine, of course, but we also need physiology. And this physiological aspect is, of course, very nicely supplemented by Pedro Valdez Sosa, and this connects us also to other brain initiatives like the Global Brain Consortium. Last but not least, Blake and Doina are 2 researchers, 2 colleagues who are very much involved in AI. They are not the only one, but they really try to use knowledge from the BigBrain to advance the architecture and the algorithm of AI. So the network becomes broader and broader, and we would say we have now about 800 researchers in 42 countries, including also Australia and China and Europe and America and South America. And we started from just 2 cities, yeah, from Montreal and Jülich, which is of course equally big, certainly. So we developed this network, and I mean, research is international, and we don't care about borders, but it was very nice to see for Ellen and me that this really also works out. So, our aim was to build a bridge between Europe and America, and we included different organizations that are supporting us, that benefit from us. We are supporting from their networks, and the Global Brain Consortium I have just mentioned, where Ellen and Pedro are very active, but we have also in particular Healthy Brain for Healthy Life, which was one of the major pillars, I would say, of support from the very beginning in Canada, and Helmholtz has it been on the Jülich side. In order to be able to grow the community, we organize different workshops, and you see that we have been at very nice places. We started the workshops earlier than the joint lab started, and we went to such beautiful places like Zadar in Croatia and Reykjavik, and we had also virtual meetings in between, so it's really a very lively, I would say, atmosphere, and I really would like to invite you also today to discuss, to participate in the panel discussion and contribute. And we can see that this type of collaboration is successful also in terms of what is being published, how the BigBrain data set is being downloaded, and you can download it from the Montreal FTP server, but also from eBrains. And there's of course some synergy, because the visibility is different in different parts of the world, and it's good to join forces, really to make the product, so to say, of research available at both sides. There are very high level and interesting publications, so I will not go into detail. Just do a Google with a BigBrain and a lot comes up. But what I find also important is that there are many publications now, or increasing numbers, that cite the BigBrain, but that are not coming from the BigBrain consortium. Yeah? So people just take it, use it, sometimes cite it well, sometimes cite it not so well, but they use the BigBrain as a tool in order to inform some kind of research. And the poster that we showed here also at the OHBM gives some examples for different research fields. We contributed to educating PhD and postdoc projects, and this is only a small, so to say, a small collection. We have a very regular and lively exchange of researchers coming from Jülich to Montreal, also a bit from Montreal to Jülich, and all of them benefited, of course, because they learned something that they would not learn when just staying at home. And I think that's a very important take-home message also for the researchers. So let me start to give a few highlights. You will see many of them, so I can quickly go, but I would like to raise your appetite for the following talks also. So BigBrain is part of the Jülich Brain Atlas, and we have just released version 3.1 with 227 cortical and subcortical regions, and we are more and more increasing mappings also of the BigBrain. So the BigBrain is one part of the brains that we have studied for the Jülich Brain Atlas, but we can map in more detail, of course, the areas. We are increasingly mapping with very high resolution. And since one brain is no brain, one could say, and variability plays a role also in the BigBrain business, we are also developing a second BigBrain, and Hartmut is here, and with Claude together, the second BigBrain is getting more and more ready also. Go to the poster. Then I would like to give an example that the BigBrain is really key in order to map some of the areas that might be so small and so tiny that you simply do not see it in MR imaging. And the hypothalamus, I would say, is one of the examples. And Alexei will give a talk later, and he has mapped the hypothalamus. And the hypothalamus is really one of the most difficult region, I would say. It's located very ventrally, which is also not nice in terms of susceptibility artifacts when you image patients, for example, or healthy subjects in that region. And the contrast is really lousy, not only in MR, but also in histology. But Alexei managed to map 23 different substructures of the hypothalamus, and they are doing completely different things. So let's see what he is doing. Another region that is also, I would say, quite nasty, this is a so-called piriform region. So the piriform region is located at the mesial surface of the brain next to the entorhinal cortex. And when you have a look, to the architecture of the piriform region, then you can see that it does not have this very easy 6-layered isocortical structures. I mean, it's a cortical region that goes to the allocortex. You have less layers, and these layers are sometimes, even on the microscope, they are very difficult to be observed. And we have no way at this moment to detect them in an automatic way. Yeah, it's even by eye, it's difficult. However, we would feel that when we do this kind of work and then train, so to say, an AI approach with it, then we would also be able to see this region. And I find this region also so interesting because it has a completely different, I mean, all the principles that we know of information processing, of supra versus infragranular layers, this is different here in this region. So the information processing is completely different. And last but not least, when we are looking to surface reconstructions, FS average brain, they all have their problems in this particular region. They are not really nice. Yeah? And having the big brain, I think we can inform also these models of surfaces to be more precise in this kind of region. We are doing a lot of AI and also in order to better understand the architecture of subcortical and allocortical region. And I would like to point your attention to a talk by Alexander Oberstrass, who was in Montreal and worked together with Jordan Kraker, and is working on the hypothalamus and integrating 3D polarized light imaging in order to better understand the internal structure of the hippocampus. And again, 3D is extremely important to understand how the hippocampus makes a connection, how it is functioning, how it is transmitting information to other brain regions. When we speak about histology, we cannot avoid talking about artifacts that are being introduced by histological processing. That's a major problem. And although the labs are well and they are doing a great work, we cannot avoid of having ruptures and all kinds of problems. And there are different possibilities, and we started already 20 years ago, I would say, to repair it. But Jan Oliver Kropp will say that there are more modern methods for removing artifacts through denoising, diffusion, probabilistic model, and repaint. And the results that we get, even on a quantitative level, shows parameters that correspond to what we see in nature. And this is extremely important. It's not just making it nice and polishing problems, but it's important to polish it in the right way and not to distort the numbers behind cell distributions. And another example coming from the big brain, this is our work with Jean-François Mangin here, is Jean-François is developing with his team methods to better align brains to each other. And this speaks to the way how we are aligning also the big brain to other brains in order to capture inter-subject variability. And folding of the brain is extremely variable and differs significantly. And it differs in a different way. So some sites are more stable, some sites are very variable. However, we would like to understand what is the relationship of microstructurally defined areas to these sites. And in order to understand this, we need to have a very precise 3D representation, and then we can study as for example, we did it here for Broca's region. On the right side, we can better understand what is the relationship indeed. And we had always these debates with Carl Zilles, with Ellen also, how much are areas related to depth of salzei. And I'm convinced, of course, it is not an arbitrary relationship. This is not the case, yeah? But the relationship is very complicated to be described. For some regions, it's very straightforward. Central sulcus, motor, somatosensory area, very easy. But when you come to Broca, how to describe it in a good way? And secondly, I mean, when you speak about connections that go from one wall of a gyrus to the other wall of a gyrus, this is another type of connectivity, as if you have, so to say, fibers that go below a sulcus. So the type of connection is completely different, and we need to understand it. I think Big Brain and Big Brains are a very nice tool to approach this. And having said that, I will hand over to Ellen, to one of the regions that was always a little bit neglected because it looks so uniform, the cerebellum.
We all get involved in collaborations of various kinds, and sometimes they work. And I'm delighted to say that the collaboration with the Jülich group has been one of the most successful collaborations that I've ever been involved in. It's been a real joy to work with the Jülich group. What I'm showing you here is just ongoing work that shows the 6-layer segmentation of the cortex from Conrad Wagstill, and that's superposed on a more recent segmentation of the cerebellum from Juan Li. As Katrin said, the cerebellum has been not given proper recognition, considering it has 4 times more neurons than the cerebrum. So I'll continue the theme of just a few highlights. We can't possibly cover the entire waterfront of all the talks that are being presented here, but just a few more highlights to whet your appetite. There are a growing number of collaborations with Jordan de Cracker and his segmentation of the hippocampal subfields and the mapping of those subfields onto a flat map, two-dimensional manifold using the hip unfold technology, and this continues to grow. Every time I raise this, people say, yes, we know all about Jordan, we've been working with him. So I guess it's good to see that that's evolving continuously. Further example of collaboration between the two groups is the work of Thomas Funk. Thomas did his PhD with me working on these data that came originally from Carl Zilles and the chemoarchitectural maps from Jülich. They were two-dimensional maps for a long time. We've all seen these pictures at various meetings over the last 20 years, and Thomas took all of that data and mapped it into 3 dimensions. So that was a tour de force of overcoming a lot of methodological errors. Thomas is here now. He after doing his PhD with me, he went on to do a postdoc with Nicola Palomero Gallagher in Jülich, and now he's a grown-up. He's a faculty researcher at the Child Mind Institute in New York with Mike Milham. Justine Hansen, she's a student with Bratislav Mizik. Justine is a force of nature. She's not here with us today, but her work is. This is the complement of the chemoarchitectural maps that you saw from Thomas Funk. This is a meta-analysis across 1,200 different data sets from 26 sources that Justine built into maps of 20 different neurotransmitter and neuroreceptor maps. Also from Bratislav's group is the so-called NeuroMaps Toolbox, which is a multimodal framework to allow mapping between the 4 major coordinate systems, annotation, ontology harmonization, and map-to-map similarity functions. This is an example, as I'll mention later, of a whole series of tools which have come out of HIBALL. Justin recently has been working on the integration of brainstem and cortical functional architectures with 58 brainstem nuclei, 400 cortical regions. She's just received the OHBM Carl Zilles Award in Integrative Neuroscience. So we're delighted for Justin. Moving on, out of the work that I've been particularly involved with Pedro Valdez Sosa, you heard me talk yesterday about the Global Brain Consortium. There are a growing number of international networks. Pedro and Jorge Bosch-Bayar have been using the big brain cortical layer structure to explore the EEG forward problem. And that work has continued as Anna's lady, Gonzalez Michens, Anna's lady, she's here. Annie did her PhD with Pedro. She's now in my lab doing a postdoc where she's exploring high-dimensional neuromass models using the big brain dataset and HIBALL as a framework within which to build a network modeling simulation environment. So what have we been providing through HIBALL? We've developed a lot of tools, some of which you've heard us mention. NeuroMaps. Atelier 3D is a three-dimensional tool for exploring gigabyte-sized data sets. That's no small feat to be able to explore the whole of the big brain in real time. Zebra, the browser to explore atlases. We had a whole session here from Timo Dickscheid and the team. So lots of tools. We also have lots of maps. The original big brain, I mentioned the cortical layer maps, psychoarchitectonic maps, 3D chemoarchitectonic maps, the Jülich brain atlas on eBrains, and coming soon, the 1 micron isotropic big brain 2. So just to fast forward for today. Yesterday we heard a tour de force from Michael Hawalitz on the Allen Brain Institute's Atlas work, the single-cell transcriptomics initiatives. We're going to hear tomorrow from Maurizio on the local activity. And today I'll introduce Michel Thibault de Schotten shortly, who's going to be our keynote speaker today. We also have the local organizing committee session to explore connectivity and clinical applications of disconnection in brain disease. And we have a panel discussion to look at challenges of big data integration. Katrin and I will give some talks on our respective infrastructures, and what's important and what we have been working a lot towards is the transparency and integration, transatlantic integration of these frameworks. There's a lot more to be discussed on that front. We have a stellar panel of participants, and that's it. We are HIBALL. It's a growing network of people. Started out with 2 sites, and now we have, as Katrin said, 800 researchers. So I think everybody enjoys coming to nice places. So that's all we're going to say, just a set of teasers to give you an idea of what's coming for the rest of the 2 days.